In this video, we're going to cover the last part of this module, talking about cell signaling and receptors. We've got a few different learning outcomes for this module. Again, this video is not going to cover all of them, but you should be familiar with them from your reading. So signal transduction involves the binding of a ligand or a primary messenger that is outside the cell to a receptor, which is inside the cell membrane. And this is going to start a signal transduction cascade that involves second messengers and can lead to a cellular response or changes in gene expression, for example. And signal cascades are important because they allow us to amplify a signal. So we can have one molecule binding to a receptor, and through the expansion of all of these signal transduction steps, we can have a large controlled cellular response. We're going to talk about two classes of receptors here. The first is our G protein coupled receptors, or GPCRs, which involve a 7-membrane spanning receptor that interacts with an intracellular G protein. And the G protein is either a monomeric or heterotrimeric. We're going to focus on these heterotrimeric G proteins. These have 3 subunits, an alpha subunit, a beta subunit, and a gamma subunit. And the alpha one is the one that binds to GDP or GTP, GTP being active, GDP being inactive. GPCRs are used throughout the body. They're used for vision, for smell, for taste, for our sympathetic and parasympathetic signaling. And again, the protein, the G protein, the part intracellular, can exist in 2 states, either the active form when GTP is bound or the inactive form. Now, what happens when it becomes activated is the heterotrimer is going to dissociate. And the G alpha, which is bound to GTP, is active and can go and do work. And then the G beta gamma, those stick together. They always exist as a dimer. So in action, when the ligand binds to our GPCR, we're going to activate our G protein, and the alpha subunit can go off and do work. And the beta and the gamma subunits will stick together. And after the alpha subunit does work, it will eventually switch out that, it'll lose that phosphate group and become GDP, and it can then reassociate with the other parts of the G protein. So GPCRs use 2 different cascades depending on the G protein that it recruits. If it uses GS, then the alpha subunit is going to start a process to make a second messenger called cyclic AMP. However, if it uses GQ, then it's going to start a process that will make a different second messenger, IP3. Now, to do this, GS is going to activate the alpha subunit of the GS protein, is going to activate an enzyme called adenylyl cyclase, which is going to make the molecule cyclic AMP. Cyclic AMP can then pass on that phosphate group until it's degraded by phosphodiesterase. And it will pass it on usually to protein kinase A, or PKA, which is a tetramer, and it has regulatory subunits that have cyclic AMP binding sites. Once cyclic AMP is bound to the regulatory sites, the catalytic spots subunits will dissociate and can go off and phosphorylate other cellular enzymes. Now, GQ here, the alpha subunit, is going to activate a different enzyme, phospholipase C, or PLC, which is going to split a membrane lipid called PIP2 into 2 components, IP3 and DAG. IP3 can go and trigger the release of calcium from its intracellular stores, while DAG can activate a different kinase, protein kinase C. Now, I know these are a lot of pathways. I suggest you actually draw these out. So if you want to pause the video and take a piece of paper and draw out these different signal transduction cascades here. Now, I said that IP3 can trigger calcium release. That's because calcium is kept under tight cellular control. So usually the amount inside the cell is very, very low, and that's because it's being either pushed out of the cell by sodium-calcium exchangers or by calcium ATPases, or it's being put into the endoplasmic reticulum by a calcium ATPase in that endoplasmic reticulum membrane. However, once it's released, it can flood into the cell, either from IP3 binding to the IP3 receptor channel in the endoplasmic reticulum, or from another receptor that is important for cardiac physiology, the ryanodine receptor. Now let's talk about a different receptor subclass, receptor tyrosine kinases. Receptor tyrosine kinases will initiate a different type of cellular response. These are single-span receptors, And when a ligand binds, here shown an example of a ligand, EGF, it's a growth factor. This is going to cause the receptors to dimerize and then autophosphorylate. So we'll add phosphate groups to all of their tyrosine residues. This will then start a more complicated cell cascade that will eventually activate the RAS protein and eventually affect MAPK, which will affect transcription factors and cause a whole host of gene changes. Now, MAPK in this pathway is involved with cellular growth, and it's important because it's often mutated in cancer. So in this video, we covered two classes of receptors and their associated signal transduction cascades. And this is going to be the end for this module.